assistants and a technician that do a lot of benchtop research and actually some of the animal work. And we have a clinical trial group, which consists of a clinical trial supervisor, a clinical trial technician. So we're going to talk about how clinical trials are done in client-owned animals, which is a little bit different from other types of research. These people are very important. And we have, Sarah's actually doing a PhD in the lab as well. So next slide, please. So what I wanted to try to accomplish in this casual atmosphere were a few objectives. First, just to get on the same page about what is regenerative medicine from our perspective as veterinarians and scientists. Secondly, where we get our inspiration for the science, which is a lot of it is from the animal kingdom and also from developmental biology. And then when you're in the area of regenerative medicine, the ultimate goal is to advance new understanding about biology, but also new therapies. And to advance and develop new therapies, you need to have a good understanding of what tools are available. So I'm going to outline for you what kind of tools are currently available for developing new therapies. Why talk about pets? I mean, that's the name of the talk. Well, it turns out that pets are integral to the whole process of developing science and developing new therapies, because they're a lot like we are. And we'll discuss what the attributes are of pets that make them particularly suitable to study and study their naturally occurring diseases that inform regenerative medicine. And lastly, we'll talk about the rationale for going ahead with clinical trials, organized clinical trials in pets that have naturally occurring disease in our hospitals, and then of course we'll open up for discussion next. So I like these definitions, which came from Mason and Dunno, a paper that came back in 2008. It's the process of replacing, engineering, or regenerating tissues or organs. And generally what we're trying to do is we're trying to normalize what goes on at the functional and structural level of tissues. The neat thing about regenerative medicine is that we're trying to address a lot of disease problems for which there are no therapies, for which therapies now are either ineffective or the patients are refractory. So keep that in mind, because there are literally millions of humans in America right now that are suffering from diseases that are refractory to any known therapy. You know, autoimmune diseases are classically refractory to a lot of different treatments, or the patients can't tolerate those treatments. Because, you know, steroids can be very, the side effects of steroids and chemo agents and immunosuppressive agents are very bad. So we're trying to address a whole area of medicine where there are no new therapies, or the therapies are crappy, or the patients don't tolerate those. So keep that in mind. Like, we're not looking to replace good, cheap therapies. We're looking to develop new therapies that can push the envelope as to what we can actually treat and make them affordable for everybody. So that's why regenerative medicine is thought to be a disruptive technology. Sort of like the flashcard was for the floppy drive. Next slide, please. So for those of you that have a love of animals, all things animals, veterinarians, zoologists, people that are enthusiastic about the animal kingdom, you'll be happy to know when you walk the beach this year, when you walk in the woods, you're coming across all sorts of animals that are really adept at regenerating their body parts. And they can perform biological feats that we could never even dream of. And so, you know, I just came back from my vacation today, and I couldn't help but, you know, look at all the sea stars and all the mollusks and all the, you know, all the hydras and all the animals out there, sea cucumbers, which we don't have in the Cape, but those type of animals that can regenerate their organs. And we're talking about the echinoderms and the octopus and squid and coelenerates. But we also have vertebrates that are also very, very capable of regenerating body parts. For example, the newts and the salamanders, which are part of the Urodele family, are very capable of regenerating entire limbs, their spinal cord, the lens of their eye, parts of their heart, and their skin, and regenerated in a way where it's a fully normal tissue. ever recognized based on its structure or function that it was ever damaged. That's something that we're not capable of doing. In fact, one of the most common things that's often talked about is how urodales and lizards, when they're faced with a predator, can run away by dropping their tail. And so they're capable of this process of releasing the tail and then regenerating it at a later time, because obviously they need their tail. There's only one mammal that can actually perform a similar trick, and that's the it's a silky mouse, and this spiny mouse, the African spiny mouse, and this mouse can actually drop its skin. So if it wants to get away from a predator, it just releases a whole bunch of skin. The predator's left with a lot of skin and hair in its mouth, and the mouse runs away. But that's the only actual mammal that we know that's capable of dropping a body part to run away. Okay, next slide. So anyways, this is where we get our inspiration. A lot of the studies that have been done on a molecular basis for regenerating limbs and eyes and skin serve as a framework for us to understand how we can, what is possible in the mammalian kingdom that isn't possible right now. Just to put things in context, humans and larger mammals have very limited capacity for true regeneration. In fact, the only real true regeneration that occurs in humans is the distal finger, the digit, the tip of the finger, and that's really only in children. And it's really only if the nail bed is left intact. Recently, in fact, last year, it was discovered that there are stem cells under your nail bed that actually regrow the tip of your finger. So if you smash your finger and you damage the nail bed, that's not going to happen. So if you're lucky enough to cut your finger in such a way that the nail bed is still intact, you'll grow back that digit. So, but there are,
Just for kids or when in adult life also you have stem cells?
It gets much more limited in adult life. Like all of these facets of regeneration, age has an impact. The exception, of course, is some of these salamanders and newts in particular you can keep, now I wouldn't really want to do this in the lab, but you can remove the lens of the eye or cut the limb off and they'll keep regrowing all through life. So they never become anything other than a juvenile. As we age, we lose all our ability to repair and regenerate organs and tissues. So yes, this is really, fingertip regeneration is really typical for children. We can regenerate our entire blood system repeatedly through life. That's a form of regeneration. And if the skin is cut badly, it's a bad wound of the skin. If you're in utero, so if you have in utero surgery, or if you're a very young child, you can actually repair that skin without any scar tissue. But after you get to be about 3 months of age, or 6 months of age, you lose that ability, and you always repair skin defects with scar tissue. Okay, so we have a lot to learn from salamanders because salamanders repair all their skin wounds without any scar tissue. And they do that by covering up the wound really quickly by the surface layer, and then they have this ways of blocking inflammation and blocking the recruitment of scar cells into that area. And that's actually the goal of all plastic surgery right now, is to actually try to recapitulate that process. So there's a lot of talk about organs that can regenerate, and then we talk about in the literature and in the late press, we talk about liver regeneration and kidney regeneration and lung regeneration. The truth is, if you cut off anything in those organs, it doesn't grow back at that site. What will happen is the tissue mass somewhere else to restore the function of that organ will start to magnify its size. So if you remove one kidney, the other kidney will go back to about 80% of the total size and capacity. You have a bigger kidney. If you cut off one liver lobe, another lobe will grow there. If you cut off, in this case, the lung, one of the left lungs of this was a mouse, this is a CT image of the mouse. You see, the lung doesn't grow back where it was cut. It actually just gets bigger in the middle. So that's not really regeneration. It's called compensatory growth. So just terminologically, we really don't regenerate anything as humans. We're really not capable of true regeneration of an organ. And that's why it's so interesting and fascinating to look at lower vertebrates and invertebrates. So the guiding principles of regenerative medicine, where we get our complete understanding of the molecular machinery that's involved and the physiology and how that's possible, are through the animal kingdom, but also during the process of development, where that happens naturally, where we form a total organism from a single cell, or from two cells. Okay. The toolbox. Okay, so having the knowledge that the animal kingdom and developmental biology gives us in terms of what is possible, how are we going to try to convert a human, a mammal that cannot, or a pet, that cannot regenerate an organ into one that possibly can? And that's the state of the art right now, is how do we actually turn us into salamanders, and actually there's a lot of the Department of Defense funding to actually support salamander research, to regrow a limb that's been damaged in the war. So the tools that are available are highly technical, but